Good morning, students. Today we'll study about red blood cell. In humans, mature RBC are oval, flexible, biconcave disc. They lack a cell nucleus and most organelles to accommodate maximum space for hemoglobin. 2.4 million new RBCs are produced per second. These cells develop in the bone marrow and circulate for about 100 to 120 days in the blood before their components are recycled by macrophages. Each circulation takes about 20 minutes. Approximately a quarter of cells in human body are RBCs. The only known vertebrate without erythrocytes are the crocodile eyes fishes. They live in very oxygen-rich cold water and transport oxygen freely dissolved in their blood. While they do not use hemoglobin anymore, remnants of hemoglobin genes can be found in their genome. Crocodile eyes fishes. Secondary functions of RBCs. Each erythrocytes undergo shear stress in constricted vessels. They release ATP, which causes the vessel wall to relax and dilate so as to promote the normal blood flow. Second, when their hemoglobin molecules are deoxygenated, erythrocytes release S-nitrosothiol, which also act to dilate vessels, thus directing more blood to the areas of body depleted of oxygen. Erythrocytes can also produce hydrogen sulfide, a signaling gas that acts to relax the blood vessels. It is believed that cardioprotective effect of garlic are due to erythrocytes converting its sulfur compounds into H2S. That is why garlic is very important. Erythrocytes also play a part in body's immune response. When lysed by pathogen such as bacteria, their hemoglobin releases free radicals, which break down the pathogen cell wall or membrane and killing it. Mammalian erythrocytes are unique among the vertebrate as they are non-nucleated in their mature form. These cells have nuclei during early phase of erythropoiesis but exclude them during development as they mature in order to provide more space for hemoglobin. In mammals, erythrocytes also lose all their cellular organelles like mitochondria, Golgi complex, endoplasmic reticulum. As a result of not containing mitochondria, these cells use none of the oxygen they transport. Instead, they produce the energy carrier ATP by the glycolysis of glucose and lactic acid fermentation on the resulting pyruvate. Because of lack of nuclei and organelle, mature RBCs do not contain DNA and cannot synthesize any RNA and consequently cannot divide and have limited repair capabilities. This also entails that no virus can evolve to target mammalian red cells. So see here how the development of RBCs is there. In the bone marrow, pluripotent stem cells they are present. They will change to burst forming unit erythroid cells, that is BFUE, under the influence of a hormone known as erythropoietin produced by the kidneys. Burst forming unit change into colony forming unit erythroid cells, that is CPU, and It is also under the influence of erythropoietin. They will change to pro-erythroblast. So this total time it takes 10 to 13 days. That is from pluripotent stem cells to BFUE cells to CFUE cells to pro-erythroblast under the influence of erythropoietin. It takes about 10 to 13 days. Then the pro-erythroblast change to erythroblast under the influence of RN. And the erythroblast change to reticulocyte again under the influence of RN, and the nucleus is removed from it and it is converted into RBCs and it is thrown into the bloodstream. This whole process will take 3 to 4 days. In this way, we can say that about 21 days they are taken for the full maturation of RBCs in the bone marrow till their release into the bloodstream. In large blood vessels, RBC sometimes occurs as stalk, flat sides next to flat side, that is known as the Rolex formation, and it occurs more often if the level of certain serum proteins are elevated, as for instance during inflammation. RBCs are also referred to as erythrocytes, as you know. They are more abundantly found blood cells as compared to WBCs and platelets. They form 45% of total blood volume. They are around 6 to 8 micrometer in size, and human body contains 4.4 to 6 million per mm cube of them. RBCs, they are, as I've told you, biconcave, lens-like, and they contain hemoglobin. They lack the nucleus. So 33% of RBCs is nothing but hemoglobin only for best carrying capacity for oxygen. Embedded within this hemoglobin molecule is iron, whose function is to transport oxygen and carbon dioxide in the blood. This hemoglobin is also responsible for biconcave shape and red color of RBC. RBCs are formed in bone marrow under the influence of hormone erythropoietin by a process known as erythropoiesis in just 7 days. On maturing in bone marrow, these cells are circulated in the blood in the body for almost 3 months before they are destroyed in the spleen and recycled by macrophages of the body. The general lifespan of RBCs is 120 days. The main function of RBCs is to transport oxygen from the lungs to other parts of the tissues and cells of the body. The hemoglobin present in the RBC is a protein which binds itself to oxygen molecule inhaled. When a person inhales oxygen from the atmosphere, it enters into the body through nostrils and reaches the lung. At the lung, hemoglobin molecule binds themselves to oxygen molecule and move to the heart. From the heart, the same oxygen carrying blood is pumped to rest of the body, that is muscles, tissues and other organ. The hemoglobin molecules then release oxygen molecule to the cells of the body. Thus, RBCs actually perform the function of transporting life-sustaining oxygen to different parts of the body. So see here in this diagram, we have got the hemoglobin molecule formed by 2 alpha and 2 beta chains, and RN is present in them, and the oxygen it binds, right? So when the oxygen is present in less amount, that is the partial pressure of oxygen is less, the oxygen is released. During the release of oxygen, 2,3-DPG plays a very important role, which is produced during glycolysis. This changes the configuration of hemoglobin molecule, and the slit, it becomes narrowed and oxygen is released. It also carries carbon dioxide. Carbon dioxide is actually formed in the cell as a result of chemical reaction taking place. This waste product is then excreted through blood plasma and RBC. While the RBC play a major role in eliminating carbon dioxide from the cells, blood plasma also accounts for small amount of carbon dioxide removal. The hemoglobin in the RBC bind the carbon dioxide molecule to form carb-amino-hemoglobin. However, unlike oxygen molecule, carbon dioxide molecule does not bind to the iron part of the hemoglobin. Instead, they combine with amino acid group on the hemoglobin peptide chain. Thus, RBC transport carbon dioxide from various cells to body and takes them to the lungs from where they are discarded by exhalation. Any factor that decreases the oxygen level in the blood, such as lung disease, anemia, increases the level of erythropoietin in the body. Erythropoietin stimulates, as I've told you, the production of RBCs, right? By stimulating the stem cells to produce more and more of RBC. 90% of erythropoietin is made in the kidney. When both kidneys are removed, or when kidney failure is there, person becomes anemic due to lack of erythropoietin. Iron, vitamin B12, folate are essential for the production of RBCs. The ratio of cell in normal blood is 600 RBC for each WBC and 40 platelets. 97% of oxygen is carried by blood from the lungs through hemoglobin. Rest 3% is dissolved in the plasma. So hemoglobin allows the blood to transport 30 to 100 times more oxygen than it is dissolved in the plasma alone. Hemoglobin combines loosely with oxygen in the lungs where the oxygen level is high and then easily releases it in the capillary where oxygen level is low. Each molecule of hemoglobin contains 4 iron atoms and each iron atom can bind with one molecule of oxygen. For a total of 4 oxygen molecule and 8 atoms of oxygen for each molecule of hemoglobin. So the iron in hemoglobin also gives blood its red color. 33% of RBCs is hemoglobin. The normal concentration of hemoglobin in the blood is 15.5 grams per deciliter of blood in man and 14 grams per deciliter of blood in woman. Besides carrying oxygen to the cells of the body, RBCs may help to remove the carbon dioxide. The carbon dioxide enters into the blood capillaries and is thrown out through the lungs. RBC is very important. RBCs contain an enzyme known as carbonic anhydrase, which helps the reaction of carbon dioxide and water to form H2CO3 plus hydrogen ion plus bicarbonate ion. This process is 5,000 times faster in RBCs because of the presence of enzyme carbonic anhydrase, which is not present in the plasma. So, the transport of carbon dioxide through bicarbonate ion is about 70% due to carbonic anhydrase enzyme. 7% of carbon dioxide is dissolved in the plasma and remaining 23% of carbon dioxide combines directly with hemoglobin to form carb-amino-hemoglobin and released in the lungs. So, regarding carbon dioxide, we can say that 70% moves through bicarbonate ion, 7% dissolved in plasma, whereas 23% forms carb-amino-hemoglobin. So, I'm thankful to www.google.com for providing us beautiful pics so that we can explain our topic well. Thank you.